let's get started. And you can see that there are several parts or sections that you're doing observation. One is close to yourself at the station page. And then the other section is here on this bench. So I'm gonna demonstrate what we're going to look at today. So first of all, you have a sheet. This is a checklist. It's a checklist for you to keep track of your observations. This is what we're going to look at today. You can also make notes. Whatever you do, this page belongs to you. You can take it with you. So let's see. First of all, on this bench, while you're working on at your own station, I will call your name to come here and then look at this. And when you come here, first you need to sanitize your hands and then start one by one. So here, the first thing, this is Firelight. I believe this is pear. So you can see the black tissue are infected. So now I'm gonna ask, is fire blight a bacterial pathogen or fungal pathogen or virus? Remember?
Virus.
Bacteria. Yeah, it's bacteria. It's Erwinia amylabra, right? So after infected, the tissue, the leaves, the buds, blossom will become dark black, and that's also how the name fire blight comes from, right? And there's another name which is shepherd's croak, because the tissue will be wilted and it's hanging. It's like shepherd's croak hanging up on the tree. So that's how the name comes from. That's fire blight, and that's the first one on your list. And the second one is, well, it's not following this order, but here, this is crown gall. And again, another bacterial disease. It is caused by which pathogen?
Agrobacterium.
Yes, Agrobacterium tumefaciens is a very important pathogen because it causes crown gall disease on plants, but also used for making transgenic plants. So there are several different samples, and this one is actually very pretty. So take a look. And then we have here is 2 plates of ergot of rye. This is a fungal disease. It's caused by Claviceps purpurea, which is an ascomycete disease, right? We probably didn't get a chance to look at this. Maybe you did, but again, let's have a look at this under the dissecting scope. And here, in the lecture, I mentioned about this. It's called corn mushroom, corn smut, right? And it's called Kutalakochi. Not sure if my pronunciation is correct, but this is made from corn smut. And I feel bad putting this one and this one together because this one is infected corn ears. And so all these black stuff, all these black stuff are corn smut, the teliospores. It's full of teliospores. So you can make a slide. I have a toolbox here. You can make a slide, tape mount or water mount, and then do observation under the microscope. You can see the teliospores. So that's that. And then here, in this tray, it's stem rust of wheat. So these are the straw. So on the straw, what can you see? What do you see on the straw?
The rust.
Rust. And what structure? What structure? Here on the straw, it's already harvested. There must be the overwinter structure or survival structure. What is that?
The teliospore?
Yeah, teliospore. Teliospores is the spore, survival spore that's produced in telia, the structure. So you can see the black structure on the straw, and you can also make a slide, make a tape mount slide. That way you can see the teliospores. And then we also have some specimens. This is stinky smut and loose smut, so you can have a look. And these two jars are armillaria species. So armillaria, what kind of disease they can cause? Armillaria. That's an oomycete disease. Armillaria is a mushroom. It's basidiomycete.
Is it root rot?
Yes, very good, Caroline. Yeah, root rot disease. So, armillaria. And this one, this jar, what is this? Can you recognize it? It's carrots. There are 2 carrots. Yeah, I'm sure when you get closer, you can recognize it. It's carrot. So the carrot is infected by root knot nematodes. So you can see a lot of knots on the root. That's root knot nematode. But with this jar, with naked eye, you can only see the knots. You can't the galls, the tumor, the galls. But you can't really see nematodes because you need a microscope to see nematodes. So that's that. And then we have here in the 2 bags, daughter. And one of them actually sampled from the previous class, the daughter. Parasitic plants, the vines, right? Remember? And we have leafy mistletoe over there. And the dwarf mistletoe here, and also in this jar. And that's parasitic plants. And this one is Haustoria. So Haustoria, the part that the parasitic plant used to connect with the host plant to get mineral water nutrients. So that's that. That's everything on the middle bench. I will call your name, and then you're gonna come and take a look at these. And now on your bench, I have pre-made slides. I have corn smut. I have stem rust of wheat. And for stem rust of wheat, there are several different structures. You see Ecia, you see Telia, maybe Uridia, and Pygmea, and you may not, it might be an older name of Pygmea, which is Spermagonia. So if you see Spermagonia, you know that it's Pygmea actually. So that's that, and I also have 2 more basidiomycetes, which is a polypore species and coprinus species. So from these 2 slides, you can see basidiospores. The red color small spores are basidiospores. So that's pre-made slides. And what else we have? We also have here. So on the back, it's I have rhizopus. So this is a rhizopus culture, and some of the rhizopus are contaminated. Actually, rhizopus is a post-harvest disease. It's very hard to get like single, like pure culture of rhizopus. So if you study rhizopus, it's hard to isolate it. If you don't study rhizopus, it's hard to eliminate rhizopus. So it's a difficult pathogen. So rhizopus, it produces sporangia on sporangial 4, and that sporangia on sporangial 4 structure look like a lollipop, and then inside of the sporangia, when it breaks, you can see lots of sporangial spores, tiny sporangial spores. So with this culture, you can make water mount, wet mount, or tape mount to make a slide and observe under the microscope. So that's that.